I open this academic ceremony in which Ms. Chloe Trayford will defend her thesis with the title "The World in a Grain of Sand, Mesoporous, Silica, Nanoparticles for Drug Delivery and Diagnostics in Regenerative Medicine." I welcome you and wish you lots of success, also, of course, with help of your parnims. I also welcome your supervisors, Professor Habibovic and Professor van Rijt, and of course the members of the opposition, whom you will meet later, and of course your family and friends on site and online. But before we start the opposition, I give you the opportunity to tell your family and friends what's the content of your thesis.
Thank you. Dear members of the Assessment Committee, my supervisors, parents, family, friends and colleagues, thank you for joining me here today. I'm going to present my PhD research, which is, as you said, the world in a grain of sand, mesoporous silica nanoparticles for drug delivery and diagnostics in regenerative medicine. So each one of us is genetically unique with distinct characteristics and needs. This means we need personalized healthcare to ensure effective disease treatments. Although most traditional healthcare is governed by one-size-fits-all approaches, there are some emerging therapies such as gene therapy and regenerative medicine that are personalized. For example, gene therapy designs synthetic genetic material based on a person's specific genome to alter the expression of proteins. And this is great because it can change cell behavior or it can treat diseases in a personalized way. Another example is regenerative medicine, which harnesses the body's natural repair process to restore and regenerate tissues. It does this usually by extracting stem cells from the patient, processing them, and then transplanting them to the disease site. This is also great because stem cells are virtually unlimited, stocks from the patient, and it's inherently personalized. But most stem cell therapies fail in clinical trials, and it's because we don't really know what happens to stem cells once they're transplanted. So do they migrate to the disease site, or do they stimulate residing cells? And this is why we need probes capable of monitoring stem cells and controlling their behavior after transplantation. So this is where size comes into play. If you think about the size of a stem cell, it's around 10 to 100 micrometers. But we need probes smaller than this that can enter the cell to deliver therapeutics to control their behavior or act as diagnostic agents. So we look to the nanoscale, and at this size range, materials have size and shape dependent properties, which we can easily tune and engineer to become effective diagnostic agents or therapeutic agents. In my thesis, we focused on mesoporous silica nanoparticles, or MSNs. This is because they are biocompatible. There have been several formulations of other types of silica nanoparticle that have been FDA approved and also used in clinical trials. They have a very high surface area because of their porosity, which means we can load diagnostic agent, like high quantities of diagnostic agents or therapeutic agents, and deliver them, and they are easily modified. For example, we can modify the core with metallic nanoparticles to enable imaging or sensing. We can also adapt the pores with charged ligands, which can improve drug capture, or even we can enlarge them to enable loading of larger biomolecules, such as genetic material. We can also adapt the chemical structure of the particle itself to instill bioactive properties or degradability. And then finally, we can functionalize the surface to improve cell uptake or enable targeting. the aim of my thesis was to develop MSNs that are able to monitor stem cells and control their behavior using gene therapeutics. So how we started was with a literature review on what's already been done in the field of theranostic MSNs. This sort of laid the groundwork for the rest of our research chapters. And then in chapter 3 and 4, we wanted to develop them as diagnostic probes. In chapter 3, we developed these imaging active nanoparticles, and in chapter 4, we wanted to assess how they did in vivo, how they could track cells in vivo. Then we moved on to developing them for therapeutics, and specifically gene therapeutics. So in chapter 5, we focused on developing them for controlled gene delivery, specifically to the mitochondria. And in chapter 6, we adapted them further. We wanted to improve their therapeutic efficacy. So, like I said, in chapter 3, we developed these imaging active nanoparticles. And we wanted not only to track stem cell location, but also their health and potency throughout regeneration. And this should give us a good idea of the regeneration mechanism with which to optimize therapies. So how we did this was doping our MSNs with gold nanoparticles and fluorescent dyes, enabling us to image by both optical coherence tomography and fluorescence imaging. We also functionalized the surface with positively charged amine groups, which should improve drug capture. And then finally, we functionalized the particle again with a reactive oxygen species fluorescent sensor. So reactive oxygen species are small molecules that their levels change depending on the health and potency of stem cells. So detecting them should give us a good idea of stem cell characteristics. What we saw is that we were able to homogeneously synthesize these particles and that they were efficiently uptaken into stem cells. We were able to monitor reactive oxygen species inside cells as well. You can see this by the increase in green fluorescence. But we not only wanted to check their characteristics in vitro, but we also wanted to check how they fared monitoring stem cells after transplantation. So in a corneal transplantation model, we transplanted our nanoparticle-labeled cells. And what we found after 10 days is that we could still image by optical coherence tomography and fluorescence imaging, and that the particles were retained in the stem cells throughout. So in summary, we were able to create these multimodal nanoparticles that were able to track and monitor stem cells, both the health and potency, and also they could be retained for long periods of time, which is promising for their use to track long-term regenerative processes. But we wanted to validate this theory of their their ability to track stem cells in vivo, as this is how it will be applied eventually. So, in a collaboration project with researchers at Maastricht University, who have developed a therapy model for encephalopathy, which is diffuse white matter injury of the brain, we were enlisted to help them detect how these stem cells work to regenerate the brain tissue. Because what they see in brain damaged mice, when these stem cells are applied to the nasal cavities, is they see cognitive repair. But they didn't understand how this works. So the aim of our collaboration was to understand where the stem cells end up in the body and to ascertain their regeneration mechanism from this. So what we did was to label these stem cells with our gold core nanoparticles. And because of the gold core, we were able to detect the amount of gold in each cell by mass spectrometry. This also meant we could quantify cell number in the tissues after these stem cells were applied. So that's what we did. After the intranasal application, we harvested the vital organs. And what we found is that in brain-damaged mice, there was a significant migration of stem cells to the brain, which we didn't see in healthy mice. We also saw that at earlier time points, there was more migration. So with this, we not only understand that migration is really important for brain regeneration, but we also find different ways in which we could optimize the model. For example, changing the dose, changing the time point of administration, or even the administration route. And this could be applied for other similar stem cell therapies, for example. So then we moved on to developing MSNs for gene therapeutics. And in chapter 5, we specifically wanted to deliver genetic material to the mitochondria. So this is because a huge problem in gene therapy at the moment is the controlled delivery of genetic material to specific intracellular locations. And the mitochondria is a big one because it has this double layer membrane which is difficult to penetrate. So what we did is, because genetic material is much larger than conventional small molecule drugs, we needed to adapt the morphology of our particles. So we enlarged the pores in one variety, and we loaded genetic material inside the pores. And in the next variety, we hollowed the core, and we added the genetic material into the core during synthesis. But in this variety, we also needed to ensure that the genetic material can be released. So we needed to, what we did is to dope the particle with degradable groups. And these groups degrade specifically in intracellular conditions. So this should give us a good way to deliver our genes specifically in cells. And what we found with our release kinetic study is that the larger pore particles showed a kind of burst release, but with the hollow particles, there was a controlled release over a period of about a week, and this was fairly specific to intracellular conditions. So this is important because we can help mitigate premature release, which can cause toxic downstream effects. We also functionalized the surface of these particles to attempt to deliver them to the mitochondria. And we chose 2 different mitochondrial targeting ligands, mitoporter and triphenylphosphine, and we adapted the surface. And then we put the particles inside cells, inside stem cells, and we found that the particles co-localized with the mitochondria, both by fluorescence imaging and electron microscopy. So in summary, we were able to synthesize these particles able to controllably release genetic material, and promising for the delivery of genetic material specifically to the mitochondria, which can be used to treat mitochondrial diseases such as muscular dystrophy or Leigh syndrome. So after this, we wanted to attempt to improve the therapeutic efficacy of these hollow core particles because we saw how cool and promising they were. So what we did was doping them with bioactive ions. And the idea of this is to introduce bioactivity, and specifically we used ions such as calcium, copper, zinc, selenium, and strontium. And this is because these ions have, they play fundamental roles in regenerative processes such as osteogenesis and angiogenesis. So being able to deliver ions at the same time as genetic material, we could potentially do combined ion and gene therapy, which could improve the therapeutic efficacy. We also doped our particles with fluorescent dyes, so we could simultaneously use them as diagnostic probes. And what we found is that by fluorescence analysis is that we were able to dope with these dyes and that there was genetic material encased in the core. And also we were able to dope our particles with high quantities of ions, as you can see by mass spectrometry. We not only wanted to see that, though, we wanted to check how biocompatible these particles were. So we put them into stem cells, and we found that they were very well uptaken, and that they were non-toxic up to high concentrations, aside from the copper-doped particles. But we could also use these for anti-cancer treatment, for example. So in summary, we effectively and efficiently doped our particles with bioactive ions, which could be used for combined ion gene therapy. So as I conclude, I'd like to highlight the key contributions of this thesis. Here we developed nanoparticles capable of uncovering stem cell behavior after transplantation. which could be used to streamline the optimization of stem cell therapies and improve their success rate. We also synthesized particles that were able to controllably and deliver genetic material to targeted locations, which could help to optimize gene therapies. And finally, we were able to synthesize all of our particles in one-prop procedures, and they were easily further adaptable. So this gives us the ability to quickly commercialize our particles for clinical use in a number of different theranostic applications. And overall, with this thesis, we move one step closer to optimizing stem cell therapies and gene therapies for improved patient health outcomes worldwide. And with that, I'd like to give some acknowledgements. First, to my supervisors, Sabine and Pamela, to my collaborators, I'm not going to name them all, to my paranymphs, Igor and Xingsen, the Nano Group, who some of you are here, and then the funding bodies who it wouldn't be possible without. So thank you. And I give the word back to the Prorector.
Thank you very much for a clear presentation. And then we start the opposition. And the first opponent is also the chairman of the assessment committee. That's Professor Honing. His expertise is analytics in system imaging at the University of Maastricht. Professor Honing.
Thank you, Mr. Prorector. Dear candidate, a lot of congratulations. That's not good English, but I'm Dutch, so that's okay. For this beautiful thesis, pushing the boundaries in making nanoparticles and using a lot of analytical tools. I will come back to that, of course. And also congratulations to the supervisory team. I started to read, and the introduction is very short, to the point, explicit, and it's not a textbook and not a tutorial, so I like it a lot. But the first 3 sentences, and I warn you, I don't like too much. And maybe you can have a look to this first 3 sentences. And I read here that you state that before X-ray we were maybe prehistoric. Now I ask you, do you remain with these statements of these 3 sentences, or do you like a fight?
A highly esteemed opponent. Thank you for your kind words and your question. I would say I would stick with my point to a degree.
Oh, okay. So we are going to fight. That's
Yes.
good. Okay. So that's I prepared for a fight. So that's going okay. Do you know a guy who's called Hippocrates?
Yes.
Okay. So that's about 2,000 years ago, and he found out that flies, who have very good sugar sensors, could measure the sugar content in urine, and they knew there was something like diabetes. So were they prehistoric?
I don't remember saying
nowadays
prehistoric in my thesis.
Sorry?
I don't remember saying prehistoric in my thesis.
No, but you say more or less relied on magic and religion. So this is not magic and this is not religion. This is...
I think I put mostly relied.
Oh, okay. Now that's a near escape. That's a near escape. So now I'm making it harder for you. Do you know, and she is also from your own country, I would say a giant in science, Florence Nightingale.
Yes.
And why do you know her?
For her work as an amazing nurse.
Oh, she was not a nurse. She was a medical doctor, and she was pushing nursery. But what was she more?
I don't know so much about
she
her.
was a mathematics specialist, and she's actually the mother of statistics in artificial intelligence, you could argue. So she was much more there, and that's already 300 years ago. So maybe later on, and if you should not disagree with me now, but maybe later on we will continue on that discussion and fight.
Okay.
But maybe good just to see how history and big thinkers have been there and will be hopefully there. And then we go from Florence Nightingale and statistics to page number 98. And on page 73, you use statistics in a proper manner with standard deviations. But then in 98, there is a calibration curve, and you just draw a line through it, and there's no statistics in it anyway. So what is this type of calibration curve? Is this a real statistical calibration?
Oh, this is a linear regression.
Yeah, and is this really a linear curve? Yeah, you make it a linear curve, but is it linear?
is where it's the scatter curve. What do you mean?
Yeah, but if you look to the first 3 points, this is absolutely not linear. You just make your curve through it.
Oh, yeah,
but,
true.
It's a sigmoidal.
It's an approximation, as you said.
Okay, yeah. And then, but you measure then fluorescence, and then I was looking to the unit, and a unit says, what is the unit? Can you help me out there?
Micromolar.
No.
Where? Oh, sorry. Arbitrary units.
Sorry?
Arbitrary units.
Arbitrary units. And what is our arbitrary units?
What are they?
Yeah, because as an analytical chemist, I measure intensity of photons or electricity. So fluorescence has an emission absorption and an emission intensity. So arbitrary units are just what you imagine should be a unit, or?
Essentially.
Okay. No, okay, so just for me to understand, so in principle...
It's a measure of intensity, as you said.
So it's a relative intensity plotted against the concentration, and you remain with the idea that it's linear, or not completely linear.
Well, I said there was a linear regression model that we put on it, but it wasn't. It's not like I'm saying the points are linear. But we put this regression on it to approximate the concentration between standards.
But the interesting thing is all the calibration curves per definition are sigmoidal and not linear. So here I think you have the beginning of a sigmoidal calibration curve because it's a response curve. But okay, that's for other discussions later on in analytical sciences. I want to go to proposition number 6, please. So if one of your paranyms can help me out there.
Continued investigation into the fine-tuning of MSN properties and their interactions with biological systems will surely lead to groundbreaking advancements.
Yeah, and again, I'm an analytical chemist, so I look to definitions a lot. And groundbreaking advancements. Groundbreaking, anyway, is pushing boundaries and going further, so that's in itself an advancement. But why do you use groundbreaking? By just optimizing some or fine-tuning some conditions?
It's just a nice hyperbolic word.
Oh, okay. And what did you want to say with this word? What did you want to mention with this? What did you want to say with this word?
Just that we move science forward, essentially.
Okay,
we break the ground of what's already been established. You could have also left it out and say, leads to advancements. Could also be, but it doesn't sound as nice.
It sounds nice, but we are scientists or not? We are exact or?
Well, the propositions are kind of inexact science, I would argue.
Okay, that's new for me, but that's a good idea. So I will keep also this type in my one-liners to the students. So it should not be scientific, but it can be whatever.
Yeah.
Oh, okay. Now we go to another word, and I saw that a few times, and you were doing that, and I find it very interesting. You add ions to your systems, and you call them bioactive ions, and that's the last question. What do you mean with bioactive?
So bioactive, essentially, they play a role in bioprocesses.
A positive role?
We would hope so, yeah. Depends on what you're trying to do.
All the ions are bioactive?
All ions are bioactive?
Yeah.
Is that what you're saying?
Yeah.
Well, I think it depends on... I mean, in general, we want to achieve something with naming them bioactive. We want to achieve
so if it's
promoting
toxic, it's
a certain
also
bioprocess.
If it's toxic, it's also bioactive. You agree?
I would say so. That's... Okay.
Very good.
I think
then at least we agree at the end of this deliberation. So thank you for the answers, and we go back to the rector.
Thank you, Professor Honing, for your opposition. Now we go to the following opponent, who's also a member of the assessment committee, and that's Dr. Albert Tazzi. This expertise is nanoscopy for nanomedicine in the Biomedical Engineering Institute in Eindhoven. And thank you for coming all the way from Eindhoven.
Thank you, Mr. Chairman. Dear candidate, I read with pleasure your thesis. It combines a few topics close to my heart, nanoparticles, materials, and also a lot of imaging. That's definitely my favorite technique. I would like to start the position asking one of your paranyms to read statement number 2, please.
The unique versatility of Mesoporous silicon nanoparticles supports extensive in-site modifications, allowing precise tailoring as diagnostic purpose in reproducible one-pot procedures.
Thank you. Totally agree with the statement. No fight there. But you make it sound very positive, and the versatility sounds positive. You can do a lot of things with it, but it also comes to the dark side that is very tempting. You start to take advantage of the versatility. You take your particle, you start to load DNA, add to dye, put a sensor, put a targeting ligands, and at the end you end up with extremely complex material. But you also claim you're interested in bringing this to the clinic, to translate, to impact patients. And by experience, say, more complex the materials, more difficult it is to translate. And especially, if you think a regulatory agency will not find it very easy to approve. So what do you think, what's your stance there? What do you think is, where is the best balance?
Esteemed opponent, thank you for your kind words and your question. Yeah, so you're right. I agree. The more complex, the harder it is to translate to the clinics. But what I would say is that with a one-pot procedure, you have less batch-to-batch variability because you're not adding multiple steps that reduce the reproducibility, and it also is harder to mass produce. Because there's so many steps and it's more expensive. So I think in general, in a one-pot procedure, I think is still a better option than the same but many steps. But as you said as well, like the more components you add in, the less it's.
So maybe let's take an example, the theranostic, that is very appealing for fundamental research. Be able to track the drug delivery carrier while it acts, of course, very appealing. Do you see a full theranostic nanoparticle in the clinic? Because then it's, again, as far as I know, there's nothing approved, and I understand also the reason, because you will need doctors at the hospital be able to do complex measurements and therapy at the same time. Do you see theranostic will make it to the clinic or be more a preclinical tool?
Sorry, I didn't understand the last little bit that you said.
Do you think theranostic will stay at the preclinical level to help discovery of new therapies, or will really make it one day in the future to have theranostic in the clinic?
Oh, yeah. So I think that was definitely the aim. I think that's also the way to effectively do personalized treatment on such a small scale that we kind of need. And I think I believe that's like the future of medicine, personally.
In how many years? 1, 10, hundreds? We talk about centuries.
Like it's probably going to be, at least a couple of decades, I would say, until that point. That there's many in the clinics. But there has been some, so that's promising, I think.
Okay, thank you. I want to move a bit more technical. You add these nanoparticles to cells and complex cells like stem cells. And can you help me to track them, to see how you imagine the voyage of these particles in these cells? For example, in chapter 3 and chapter 5, but let's start with chapter 3. What happens to these particles? Where are they located in the cells with their cargo over time? So how do you imagine? Because you don't fully discuss that. You just show they are inside the cell. Where are they? When? At which time point? Can you elaborate more?
Yeah. Okay. So we did for the first project with the imaging particle. We did see that a lot of them collected near the nucleus, potentially in like the Golgi apparatus, I think it's called, like collecting there. And so I think it depends really on the size of the nanoparticle, because in this project they were larger, they were 200 nanometers compared to other projects. How
did you arrive to assuming there's the Golgi? Do you have evidence there in the Golgi?
I don't, I'm just like assuming that they're doing that, but I don't know.
How do you imagine getting there? How do they reach the Golgi?
How do they reach it?
Yeah.
Just getting stuck in all the lipids bits.
So you think they fuse with the membrane?
Of the Golgi?
Well,
I'm
outside there to reach the Golgi. So there's a trip. Now it's Eindhoven to Maastricht.
Oh, how do they penetrate the membrane itself?
What's
of the
in between?
stem cells?
I knew I took my train from Eindhoven, I stopped in Roermond and then arrived in Maastricht. Where steps do they take?
Okay. Yeah. So like I said, it depends on the size and the surface properties of the particle. So in the first project, we had larger size that had amine groups on the surface, and these, we think it was more likely by penocytosis. We didn't do any endocytosis assays, but it's more likely compared to other similar studies. Whereas later with the smaller particles, I think it could just be endocytosis by ATP using mechanisms. I don't know what.
Okay, so you think in chapter 3 part want to do endocytosis and reach the Golgi? Can they get out of the cell?
How do they get out?
Can they?
We didn't get that far. We just saw that they were retained for this amount of time that we did our experiments. In general, I think the longest studies that have been seen is that they've been retained for about a month or even longer, even in vivo. So like they're quite well retained in the cell.
Okay.
Yeah, but it depends also, like in our later chapters, so 5 and 6, they degrade, so they're going to be excreted in little fragments.
And if you compare to the particle in chapter 5, they have to reach the mitochondria, same questions. How do they reach the mitochondria? How do you think that happens?
So the surface ligands, so TPP or mitoporter, but I know more about TPP, it's very highly positive. And there's a membrane potential difference, so that's the kind of driving force of them to enter the mitochondrial matrix. I don't know details of that because we didn't do the specific assays, but that's kind of what's been researched with TPP specifically.
I agree, but to reach the mitochondria, they come from endosome to mitochondria directly?
No, they have to burst the endosome for sure. We need endosomal escape.
Do you think they have sufficient endosome escape? Because that's a bottleneck of pretty much every oligonucleotide therapy, and it's where all 90, we talk about typically 95% of the particles don't do endosome escape. Do you think you have very significant endosome escape or not?
To obviously confirm that, we need to do further studies, but we did see on TEM, there were at later time points, or for TPP specifically, we did see that they didn't have this kind of vesicle around them, whereas some of the other particles did without the functionalization. We saw this kind of vesicle around them, so we could see the difference. And I think in that sense, we can kind of have this rudimentary idea that they do escape the endosomes. But obviously, like I said, we need to do further tests.
Okay. I'm not sure if I ask Mr. Chairman if I have more time or it's better to... I think my time's up. Thank you very much for your answers.
Thank you, Dr. Albertazzi, for your opposition. We come now to the following opponent, that's Dr. Cucinotta, and his expertise is supramolecular photochemistry in the School of Natural and Environmental Sciences in Newcastle University. That is in the UK, and thank you for coming all the way from the UK. The University of Maastricht is always glad to have people from abroad. The floor is yours.
Thank you very much. I would like to start by saying that it was a genuine pleasure reading your thesis. I think this is a nice piece of work, and congratulations for getting pretty much all of it published in high-quality, journals. So well done. I'd like to start by addressing one of the points that you've made in the proposition number 5. So if one of you panelists could please read that for me. Thanks.
Encapsulating oligonucleotide in the core of MSN protects them from degradation, premature release, and enables cell uptake, positioning them as next-generation gene vectors in medicine.
Sorry, I meant number 5.
Sorry.
Yeah.
Adjusting MSN degradation rate and targeting efficiency represent promising strategies for controlled organelle-specific drug release on the road to effective therapeutics.
Thank you very much. Didn't want to interrupt your proposition 4. That's interesting. I might come back to that later. But to address one of the points you've made, you said that adjusting the degradation rate of the Mesoporous silica nanoparticles is a promising strategy. Could you expand on what exact strategies you've used in your work? You've touched on several strategies and the use of several organoalkoxysilanes. Could you expand on that? Describe what silence you've used and how each of them had an effect on the degradation rate and how the degradation mechanism works.
Esteemed opponent, thank you for your kind words and your question. So yeah, we mostly used BTES, which is a disulfide bridged organosiloxane. And this is then reducible by glutathione. And so glutathione is present in the cells in very high quantities, like 10 millimolar, I think, compared to 1 to 10 millimolar compared to outside the cell, which is 10 micromolar. So it's a real 1,000 times difference, which is why we chose BTES, because then we know that this can degrade in intracellular conditions. The other strategy was just to dope with ions. So the last chapter, we just doped with bioactive ions. And this has been shown in a lot of previous research, for example, with, what are they called? Bioglass. They degrade in a much higher rate than normal MSNs because of the defects that are caused by the ions in the structure. So yeah, those are the two, but that's A-specific. That's not going to be intracellular specific, for example.
Okay. Well, you've mentioned the use of different ions in the last chapter. Preceded me. I was going to ask that at last. But how does the use of different ions affect the fluorescence output later? Because ultimately you use the fluorescent sensors or the dyes to do the imaging. So how does using different cations have an effect on that, if you have explored that?
So it's something we didn't explore, the mechanism, the effect mechanism, but we did obviously. The problem we had was that the more we dope into the silica matrix, the less of everything gets doped, because there's like a competition, a steric competition with all the different components. So in general, what we saw is that there was lower fluorescence with particles that were more highly doped with ions. But we kind of expected that from just this competition effect. But it could also be a result of quenching or something, for example.
Yeah, that's the most likely pathway for the fluorescence decrease. Okay, I'd like to move now to one of the mechanisms you describe in chapter 3, right? You describe the effect of using different concentrations on the sensing mechanism of the reactive oxygen species. Can you expand on that and also explain how the sensing works? So mechanistically, how do these reactive oxygen species get sensed or detected?
So essentially the reactive oxygen species sensor gets oxidized by the reactive oxygen species, which changes the fluorescent output. So at higher concentrations, we expect higher fluorescence. And that's higher concentrations of reactive oxygen species, I mean. So that's what you see in, I guess you're on page 83 or something.
Yep.
Yeah, so that's what you see there. And we had obviously different, depending on the way the fluorescent dyes were functionalized into the matrix, it changed the fluorescence intensity output, and I think that's to do with the keeping the dye in a kind of
It's in a specific conformation.
Yeah, exactly. Like what you said.
Right. So is it important to functionalize the particles on a specific location to attach the dye, or does it not matter? Because there's different active sites or sites that can offer functionalization opportunities. The outer coating, the inner surface. Which one is the most suitable for attaching the sensors?
So obviously we did these 4 different techniques, and I think what we found was, well, what I think is the most promising is to attach the fluorescent sensor. And I think this is more from, because with all the different ways in which we functionalize, we saw different outputs. And also this changed depending on the analysis method. So there's not like a one that stands out necessarily that's, I think, better. But in general, I think the best is to attach it to the surface, because then it's not leached and we don't cause potential toxicity in the cell, and we can also sense reactive oxygen species at specific locations as opposed to throughout the whole cell, which is less helpful and informative.
Okay. And how do the encapsulation methods, the different encapsulation methods, affect the sensing efficiency? Because you describe different ways of encapsulating and making this, these particles. So I think you described 4 different systems. So
how does it affect?
Yeah, how does it affect?
So, well, we just saw different fluorescence increases depending on the different methods of functionalization. But it was also specific to, like I said, to the analysis method. And I think that's to do with how they're differently measured. So, for example, with flow cytometry, we saw that just encapsulating them and not attaching them was better, because I think because we're detecting the fluorescence in the whole cell. Whereas with things like microscopy, we saw that the attached one's better, and I think that's because it's measuring the fluorescence at a certain plane.
Yeah.
So, yeah.
So do you think it depends also on the localization of the sensing, where you're looking at?
Yeah, exactly, yeah, I think so.
Okay. I suppose I'm almost up with time. Thank you.
Thank you for your opposition, Dr. Cucinotta. Now we come to the following opponent, who is also a member of the assessment committee. That's Professor Piech. His expertise is functional and interactive polymers in the Technical and Macromolecular Chemistry Institute in Aachen, and also in affiliation with Maastricht. And today he has also accepted the role of secretary of this committee, for which I'm very grateful.
So, dear candidate, I would like to congratulate you, and it was my big pleasure to read your thesis. A really impressive work. My first question is related to chapter 3, where you describe the synthesis of core shell particles. And reading your thesis, I noticed that you refer to Sturba method, right? So you start with gold cores, and then you coat them with silica shells. So, and you show a very nice scheme 1 on page 30, I think 64, where you show that how this works. So my first question is, can you explain how this shell growth occurs in your system, and why each gold core is coated uniformly?
Highly esteemed opponent, thank you for your kind words and your question. So, which page were you talking about, sorry?
I think it's 64.
60. Okay. I don't have it. But I will say that, so essentially the gold nanoparticles, we want to make them charged at the surface. So that's what we did. We functionalized the surface with citrate and then CTAB eventually. And this is acts as a nucleation point for the silica to start to hydrolyze and condense. So that's how it works.
So that means you assume that the condensation and formation of cross-link structures or silica occurs on the surface and not in a solvent?
I think it's depending on the molar ratios of everything that you put in. And that's also what we found there was a lot of failed synthesis. Because it's a very temperamental thing. You have to have very specific molar ratios, as you know, I guess. But then at that perfect ratio, then you can have this nucleation specifically at the surface. And it also depends on the solvent. We found, like, with changing the dielectric constants, you have like the silica kind of grows like a tube rather than like a sphere.
You have indeed very impressive TM images. So,
yeah.
And I was impressed by this highly uniform structures. You call your nanoparticles, or this, you call them mesoporous silica. So, what is actually the size of the pores? And how is there any possibility to control this parameter by chemistry?
Yeah, so as we synthesize them with just normal surfactants, so we use CTAB, the pores should be around 6 nanometer. And mesoporous means between, I think, 2 to 50 nanometer. So you can have mesopores within that whole range of diameter. But we mostly synthesize around 6 nanometer. You can enlarge them, and that's what we did in chapter, is it 5? Yeah. And this is by adding in another group that makes the vesicles, so the structure of the template larger, essentially.
Yeah, so thank you. So I also noticed that you used also functional silica precursors to integrate some specific groups, tiles and primary amines.
Yeah.
And so what was, my question is, can you really quantify the concentration of the content of these reactive groups on the surface or inside of the silica shell?
So it's not something we tried personally, but it is something we have tried in a group with different methods. We tried by fluorescence, and we tried to attach fluorescent dyes and check the difference in fluorescence. But this was not very reliable, so it is difficult to quantify. But we didn't really need to do it for our reasons. We just needed to know that they were there mostly and that it had an effect.
Okay, so now I have a question to chapter 5, and on page 127, you talk about release of oligonucleotides, which were encapsulated in the silica structures, and at the same time you comment on degradation. So apparently your carriers degrade and you have the release. So
yeah.
My question is how these two processes are connected or interconnected. Is there any possibility to understand what is the mechanism of oligonucleotide release? Is it the passive diffusion, or is it really active degradation of the carrier, or maybe both?
Yeah, no, I think it's both. I think as the carrier degrades, you have larger holes and larger, it's less structurally sound, so you have more areas with which the oligonucleotides can escape. And I think they just get released by diffusion. That being said, I also think they probably associate again with the silica surface because of their charge. But eventually, obviously, this is just given away because the silica degrades so much that it's no longer there.
So you also show very nice comparison. I think it's figure 3 in chapter 5, where you show TM images of your nanoparticles after different degradation times.
Oh yeah.
And you compare particles which had this disulfide linkages, or with addition of glutathione with and without.
Yeah.
Looking at these images, I do not really see much difference. You expect glutathione to act as a trigger for the degradation. But it seems that even after 7 days, some structures are still there.
Yeah. Sorry.
Can you comment on that?
Yeah, I can. So I think, like you said, there's not so much difference we could see by TEM, but this was also, I think, to do with the measurement that we had, because I was spinning down, like centrifuging what we had and analysing that, as opposed to taking from the solution, which is what I should have done. But I think the more reliable thing to look at is the release study that we did. And so I think it can't really be released if it's not degraded, because it won't, it can't be, can't.
Okay. Is there any alternative analytical method which can be used to follow the size of the carrier as a function of time? You mentioned that it's indeed true. If you take a sample, put it on a grid, dry it, you have a lot of, let's say, possibilities for some artifacts. Is there any technique which you can use in solution?
Yeah, so I think we could and should use dynamic light scattering.
Excellent.
We just keep it in solution throughout, and then it's always in solution as we measure, which is the more reliable thing to do, rather than sort of have these drying effects that we had for TEM, for example.
Yeah, thank you for answering my questions.
Thank you for your opposition, Professor Piech. Now we come to the following opponent, who is also a member of the assessment committee. That's Professor Balmagor. Expertise is experimental orthopedics and trauma surgery at the Department of Orthopedic Trauma and Reconstructive Surgery in the University of Aachen, and also in affiliation with the University of Maastricht. Professor Balmagor.
Thank you very much. Dear candidate, first of all, congratulations for a beautiful thesis. Also to your supervisory committee. It takes me back to my own PhD thesis, which was also in particulate system, all the chapters, so I enjoy very much reading your thesis. And I have one question, as you can expect, overall, chapter 5, chapter 6, in your gene therapy approaches. It was very nicely also mentioned in your presentation. We mostly do that today with lipoplexes and liposome systems, for instance, avoiding the use of materials in the context of a particulate system. So what will be the advantages of using particulate systems for gene therapy?
Highly esteemed opponent, thank you for your kind words and your question. Yeah, so I think that the solid systems, or like particulate systems, I like them better. No offense. But I think because they're more stable, basically. I think with the liposomes, they're hard to synthesize homogeneously. I know you can do it, but yeah, it's hard. I know there's like several steps, and it's harder to do, and they're also more stable. So, sorry, the ones that the silica particles are more stable because you can store them for long periods of time. You can't really store liposomes very well. So you really have to synthesize them at the point of care, which is not very. It's just not very viable, I guess. And when you want to operate on someone straight away, or yeah.
Very good. Very good. No offense taken. I like biomaterials sufficiently enough to agree partially with you. So we can store a little bit longer than a few days liposomes indeed, but I agree with you that the material provides extra protection to the complexes. It also, I enjoy very much the complexity of your systems, very much, and I agree with them, and I will like to take you then to how this complexity also brings limitations. It was a little bit discussed already in terms of regulatory limitations and so on. How would you circumvent this? So we have in your whole thesis different type of MSNs with porous, without, hollowed, functionalization, and all those steps are beautiful, brings a lot of good properties, but also increased complexity. if you will need, could you minimize the complexity of your system to make it robuster in terms of straightforwardness, regulatory speaking, or application?
I think so. I think if we didn't need them for diagnostics as well, we could just mitigate adding fluorescent dyes. But I think in general, like I said before, I think it's having a one-pot synthesis is doing a lot of the work. But yeah, I think taking out some of the things you don't need for a specific therapy, like if we were just wanting to deliver genes to the mitochondria, we wouldn't have fluorescent dyes. We can just put mitochondrial targeting agents and the particle. So yeah, that's what I would say.
Very good, very good. So moving a little bit into chapter 5, we have there very clearly the comparison, which I like also very much, between the hollow system, where you obviously entrapped inside the hollow nanoparticles, and the system where you have basically the surface available for loading your plasmids, your DNAs, your nucleic acids. And we discussed it already a little bit, that the hollow system depends on the degradation of the shell, indeed. How do you think the degradation of the shell can be controlled by the cellular, intracellular components? Because if you say it yourself minutes earlier, if the shell doesn't degrade, then the cargo will never release. Mm-hmm. So we are, at the end, depending on the cell and how specific that can be from cell to cell to degrade the shell.
Mm-hmm. So your question was, how can we alter the degradation rate? Yeah. So I think there's also other dopants we can use that are more sensitive if we wanted to increase the sensitivity. For example, you can do, it's like 2 disulfide bridges together. I've forgotten what it's called. But we can use that, and that's more sensitive. And there's other dopants we can use for one. And then also, like you said, the cell choice matters, because in different cells there's more or less glutathione, and that's what we were focusing on anyway. So in stem cells, there's much more glutathione than there is in
that's exactly the direction I wanted to go.
Okay.
So
yeah.
Would you then say that by selecting the type of cell, the different cellular system, would you need to tailor down the shell of your hollow nanoparticles a little bit to make it cell-specific to the degradation process to occur?
Yeah, that's a really cool idea. Just by changing the ratio of dopants that you use, I think you could achieve different degradation rates for sure.
Very good. Excellent. So I also like it very much in chapter 6, the use of ions to dope your system. And indeed, you mentioned specifically that one of the aims to use this was to increase degradation and biocompatibility. Mm-hmm. Wouldn't it be easier to just change the matrix of the nanoparticles to increase degradation and biocompatibility with other polymers?
With other polymers?
With other polymers. It would be easier not to use MSNs and to use chitosan nanoparticles.
Oh, I see. Mm-hmm. Like to use polymers or something else. I don't know about the ease. I mean, I think the ease of the synthesis was pretty easy. It was just all in one pot. It was all in mild conditions. I think the ease was not really a problem. And I think we chose this material because there's already been formulations that have FDA approval, so it's kind of halfway on the way to being approved, like our particles being approved, I mean. So, okay.
You may finish your answer, you may also say enough is enough.
Oh, sorry, I forgot the question. There's too much going on. Enough is enough.
Okay, thank you. Thank you, Professor Balmacher, for your opposition. And Ms. Trayford, the time appointed for the defense of your thesis has now passed, and the degree committee will now withdraw to discuss the quality of your thesis and the way you have defended it. And I request that you and your company await the results of our deliberations and our return in this room. And with that, I suspend this meeting.
The PhD defense has now ended. The degree committee will debate the candidate's performance behind closed doors. This process usually takes about 10 minutes.
Long road to the south side. 7 miles in my rearview mirror, I know what it felt like. My goal's only getting clearer, east side to the west side. No place like home, if it's questions that you've got, go the extra mile and die. On the line, I know you know that I can't read your mind. Years can't fly by now, don't waste all your time. Cause I'll go the extra mile. Long road to the south side, 7 miles in my rearview mirror, I know what it felt like. My goal's only getting clearer, east side to the west side. No place like home, if it's questions that you've got, go the extra mile and die. On the dash, got my laces tied. Long road, I don't waste no time. Break rules because fate decides. With the team and we chase the light. I make a move, fall down, shake it off. I hate to lose, bad branch, break it off. No room for negativity, praise and love. Prepare for deep part cause we're taking off. Take the mileage, traveling so far from home. Gathering knowledge, other connections. I put the time in to get everything I ever hoped for. It's a challenge and we all in. It's a better future that we go for. And even though these nights seem long, I know that I am never on my own. Know you've won the battle once you've grown. South side, 10 miles in my rearview mirror, I know what it felt like. My goal's only getting clearer, east side to the west side. No place like home, if it's questions that you've got, go the extra mile and die. Long road to the south side, 10 miles in my rearview mirror, I know what it felt like. My goal's only getting clearer, east side to the west side. No place like home, if it's questions that you've got, go the extra mile and die. Years can't fly by now, don't waste all your time. Cause I'll go the extra mile.
I reopen this academic ceremony. Ms. Trayford, the degree committee here present has assessed the quality of your thesis and the way you defended it. And in view of its positive verdict and taking into account your previous qualifications, the degree committee has decided to grant you the degree of doctor. And Professor van der Rijt is authorized to confer upon you this academic distinction in accordance with Dutch university custom. I invite your supervisor to now take the floor.
Do you promise to work in accordance with the principles of scientific integrity at all times, to be careful and honest, transparent, independent, and responsible? By the authority vested in us by law and in conformity with the decision of the committee here present, I hereby confer upon you, Chloe Trayford, the degree of doctor and grant you all rights attached by custom and law. As evidence of this, I now present you with the degree certificate signed by the rector, the secretary, and the other members of the committee and affixed with the official seal of the university. Dear Dr. Chloe Trayford, it is my great honor and privilege to be the first to officially address you as doctor and congratulate you on this incredible milestone. Today, we celebrate not only the culmination of your years of dedication, hard work, and perseverance, but also the unique contributions you've made to science and our group. You joined our lab to work in the Top Vision project, a collaboration between our group and the ophthalmology department, where our goal was to design nanoparticles capable of tracking stem cells on the cornea. This work aimed to uncover new insights into the stem cell-mediated tissue regeneration of the eye. And this work led to several other collaborations. As it turns out, tools to study stem cell distribution in vivo settings are of great interest in the field. For example, we collaborated with the lab of Cora Nijboer in Utrecht, who work on stem cell therapy for the treatment of brain damage in preterm babies. While their application and administration route were entirely different from our corneal studies, our nanoparticles provided critical insights. We were able to show for the first time that these stem cells applied through nasal drops could migrate to specific brain regions, significantly advancing their understanding of how stem cells repair brain damage. This collaboration is still ongoing and is now being carried out by another PhD student, Lotte. As a chemist at heart, you were truly in your element when designing and optimizing nanoparticles. I remember how you enthusiastically supervised master student Alyssa, embarking on the challenge of developing nanoparticles that could not only enable imaging, but also carry gene therapy tools. Over the course of our many progress meetings, we evaluated countless particle designs, often having to make tough decisions about which ones to pursue further. This work laid the foundation for yet another enduring collaboration, this time with the toxicogenomics department at Maastricht University. It also paved the way for the work for future PhD students, Nick and Jen, who are now expanding on this project as part of their thesis. So your many contributions have not only driven our research forward and resulted in this nice thesis that is lying in front of us today, but has also set the stage for new collaborations and opportunities that will continue to grow. For that, I'm deeply grateful. Next to your collaborative spirit, you are generous, resilient, a bit unconventional, and also someone who always tries to look at the bright side of things. You are always ready to lend a hand or share a laugh. Your presence has made the lab a brighter place. I admire your way of approaching things with humor, and while we worked hard together to get you to the finish line, we also found time to enjoy the journey. Thank you for that. Beyond your research, you have demonstrated talent for teaching. You have a gift for connecting with students and guiding them through their own learning journeys. You can break down complex concepts, for example, during the student interviews that you gave, and make them approachable. This demonstrates that you are a great educator. Though you've expressed uncertainty about your next steps, I believe your talent for teaching and mentoring will be a strength wherever your path leads you. Of course, no PhD journey is without its challenges. For you, presenting your work has been an area of growth. While your natural conversation skills shine in one-to-one settings, presenting at larger audiences has sometimes felt more daunting. Despite this, I've seen your confidence and skill grow over the years, which was also demonstrated today. One of the ways you've made your presentations stand out and made them your own is through the creative and engaging animations that you designed to explain the concepts behind your studies. You have shown resilience throughout your PhD journey. It was not always easy, with COVID shutdowns causing delays in lab work, experiments that kept failing, or nanoparticles that could not be reproduced. This, in addition to several moves around Maastricht, writing up the thesis while dealing with additional moves and life decisions in the final stretch of your PhD journey, was challenging. But you made it to the end. And as we celebrate your achievements today, I want to acknowledge not only the beautifully written thesis you have produced, but also the publications and numerous contributions to other papers that stand as a testament to your hard work and dedication. You should be incredibly proud of all that you have achieved. It's been a joy to have you in the group, and I'm excited to see where your journey takes you next. Wherever you go, I know you will bring the same kindness, curiosity, and determination that have defined your time here. Congratulations once again, Dr. Chloe Trayford. You have earned this moment. Before I conclude, I want to express my gratitude also to Pamela, as second supervisor. Thank you for your feedback and guidance that helped shape the research. I also want to thank our collaborators, Florence van Tienen, Bert Smeets, Rudy Nuit, Vanessa Lapointe, Cora Nijboer, and as well the Assessment and Promotion Committee for their contributions and presence here today. Thank you, Maarten, for being the chair of this assessment committee here today as a fellow chemist from Maastricht University. Thank you, Lorenzo, for our nice scientific discussions also over the years, and hopefully we can continue to work together in the future. Thank you, Fabio, for coming all the way over from the UK. It's been great to getting to know you today and also some of your nice work that you do. Thank you, André, also for being able to come. I know initially you wanted to be here online, so I really appreciate to have you here, and hopefully we can continue our discussions later. And finally, thank you, Elisabeth. Hopefully we can also work together on the gene therapy tools that Chloe started to develop in our group. Dear Chloe, congratulations once again on this extraordinary milestone. This day is not just a celebration of your success, but also a moment to acknowledge the support of your family and friends, both here and online, who have been with you every step of the way. I'm confident that you have a bright future ahead of you, filled with exciting opportunities and achievements, and I wish you all the best, both in your career and personal life. Enjoy this well-deserved celebration. You've earned it.
Thank you, Professor van Rijt, for a beautiful laudation. And dear Dr. Trayford, also on behalf of the University of Maastricht, I congratulate you with the degree you have acquired. And I would like to extend my congratulations, of course, to your paranymph, who did great work, and of course to your supervisors, Professor Rijt and Professor Habibovic. I also extend my congratulations to family, friends, and colleagues here present and online. And I hope you can really celebrate this most important day, one of the most important days in an academic career. Having said that, I thank the members of the opposition for their work. I also thank the PEDEL and the technical support team, because without their help we would not have this great meeting, which is already also online. I'm almost ready to close this session now. What we are going to do with the young doctor and the paranymphs, we're going to make a picture at the foot of the stairs, so the photographer can follow us. And when we have left the room on the way to the stairs, I ask you to already go to the reception room where you can have a drink or something like that. And then if you wonder when you can congratulate a young doctor, that's quite easy. When we enter, you follow us. We come first, and then you follow. Having said that, I close the meeting.
Please all rise. Academics, please bring the degree. Supervisors, please come. Professor Warning, you the rest.